Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on conducting an experiment to prove digestion occurs in the mouth. In our last lesson, we studied the final stages of the digestive system. We learned about digestion in the small intestine and the way in which food particles are transported to different areas in the body. We also looked at the role of the large intestine, as well as the liver and pancreas in the digestive system. Today, we will investigate an experiment that proves digestion begins in the mouth. We will outline the procedure of the experiment, as well as review the role of the mouth in digestion. Let us begin, students. Let us get started with an activity. Students, choose a partner and then explain the role of the teeth, salivary glands, and tongue during digestion.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Welcome back, everyone. As we have learned, once food enters the mouth, digestion begins. Food is bitten, chewed, and ground into small pieces. As the food is torn apart by the teeth, it is mixed with saliva. Saliva is a liquid that contains enzymes which break down carbohydrates. The tongue moves food around the mouth during chewing and mixes it with saliva. Then the tongue rolls the food into a small sphere called a bolus. The tongue pushes the bolus towards the back of the mouth, and a reflex action temporarily shuts off the airway, flicks the tongue upward, and sends the bolus into the esophagus. Powerful muscle contractions called peristalsis force the bolus down until it arrives in the stomach. Was your answer similar, students? Enzymes are essential to the process of digestion. In fact, enzymes control and maintain the pace of hundreds of reactions that occur in the body. Enzymes are made of proteins and are specifically tasked to catalyze a particular reaction in the body. Hundreds of reactions occur in the body at all times, and enzymes ensure that the reactions take place in a rapid and controlled fashion. Intracellular enzymes work inside of cells, while extracellular enzymes are secreted into organs, such as the gut, where they catalyze certain reactions. Digestive enzymes are extracellular. They work outside of cells in the lumen of the gut. Amylase is an enzyme that is made in the salivary gland, in the mouth and pancreas. It works to break down starch to the sugar maltose. This reaction can be observed in an experiment to indicate how quickly enzymes work. This experiment requires 3 solutions: an amylase solution, a starch solution, and an iodine solution. To begin, drops of the iodine solution are placed on 2 spotting tiles. Next, 3 test tubes are gathered. 2 are partly filled with the starch solution, and 1 is labeled starch, while the other is labeled starch and amylase. The 3rd test tube is filled with amylase solution. The 3 test tubes are placed in 30-degree Celsius water for 5 minutes. Once the temperatures equilibrate, an equal portion of the amylase solution is measured out and added to the test tube labeled starch and amylase. A timer is set, and a small sample of the starch and amylase solution is immediately taken and added to the first drop of iodine on the spotting tile. Samples are taken and color changes are recorded every 30 seconds for 10 minutes. A record is kept of the time the samples were taken from the solution and the color of the iodine. A table like the one you see on the screen could be used to keep track of the observations. At the end of 10 minutes, a sample of the simple starch solution is added to the spotting tile and compared with a sample from the starch and amylase solution. This comparison will give the person conducting the experiment the opportunity to confirm any changes that took place are due to the enzyme, rather than the temperature of the solutions. This experiment can be repeated with both mixtures kept at a range of different temperatures. If the iodine on the spotting tile turns black or purple in color, it indicates that starch is present. If there is no color reaction and the iodine remains light brown in color, starch is absent. Benedict's reagent can be applied to test for the presence of reducing sugar. Equal parts of the starch solution are combined with Benedict's reagent in one test tube. Equal parts of the starch and amylase solution are combined with Benedict's reagent in another test tube. Both test tubes are heated in a water bath for 1 minute. The results of the color of the solution are immediately observed and recorded. If the color of the solution is red, there is a high concentration of sugar. If the solution is orange or yellow, there is a medium concentration of sugar. If the solution is green, there is a low concentration of sugar. If the solution has no color reaction and remains blue, sugar is absent. Students, let us do one last activity before we conclude our lesson for today. Please answer the following questions. What are enzymes made from? What is the main function of enzymes? What is the name of the enzyme that is made in the salivary gland and pancreas? In the iodine test, what color does the iodine become if starch is present? In Benedict's reagent, what color does the solution become if sugar is present?
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, class. I am sure that was a simple activity for you. Enzymes are made from protein. The main function of enzymes is to act as catalysts. Amylase is the enzyme that is made in the salivary gland and pancreas. The iodine will turn blue-black if starch is present in the solution. Benedict's reagent will turn red if the solution contains sugar. How did you do? If you wrote down the correct answers, great work. We have reached the end of our lesson. Today, we learned about a simple experiment that shows how enzymes start the process of digestion in the mouth. Until next lesson, thank you, teacher. Thank you, students.